Hi, my name is Eric Scott, and I'm a postdoc at University of Florida. And I'm going to talk to you about some work I did as part of my PhD at Tufts University in collaboration with Elizabeth Krohn. And it's about using supervised and unsupervised analyses of multivariate data in ecology. So with the ecological data revolution, we are often getting lots of data from things like community science, remote sensing, metabolomics, eDNA, and other sources. And with multivariate data like this, it's common to have more variables and observations, or to have highly correlated predictors, which means that we need to often use some specialized statistics to deal with this. But I'm not going to talk about this sort of nitty-gritty choice of what test to use for your particular data set, but rather a more fundamental decision in your analysis of whether to use an unsupervised or supervised approach, a decision that I think is often neglected in ecology. So what do these mean? Well, an unsupervised approach is one that looks for main axes of variation in your data. So in this example here, we have a hypothetical data set with 3 variables, and we have our observations as points, and in this 3-dimensional cloud here. And an unsupervised analysis is going to look for an axis that goes through the spread of the data, finding the maximum r squared x, which is the proportion of variation in the predictors that's explained. And in this variable, or in this data set, we have a response variable. It's this color, blue or red, and it is agnostic to that response. It doesn't care about blue and red, which is why in this example, blue and red are not really well separated along that axis. This is also sometimes called an unconstrained approach. A supervised analysis, on the other hand, is one that finds axes that explain the response. And so we get a very different axis in this example because the difference between the red and the blue is not along the spread of the data. So this is maximizing the r squared y. This is the proportion of variation in the response explained. This is actually just the traditional r squared, but I'm calling it r squared y to differentiate it from this proportion of variation in predictors explained, the r squared x. And this is a constrained analysis, it's also called. And these approaches answer fundamentally different questions. It's not about which of these is appropriate for your data set. It's about which of these is appropriate for the question you have. So to illustrate this, I want to go through a case study. This data was originally published in New Phytologist by Muir et al. They looked at 16 solanum species, a few of which are pictured to the right, and measured in them 9 leaf traits, as well as the mean annual precipitation of their native habitats, which I'm going to call precipitation niche. And in this paper, they were interested in something called the leaf economic spectrum. This is an axis of variation in plant traits going from resource acquisitive to resource conservative, and it's something that seems to be found within many different groups of plants and across groups of plants at different scales. So we used these data, which are published on Data Dryad, to answer 2 distinct questions. First, is the position along the leaf economic spectrum related to precipitation niche? And we call this an unsupervised question because it requires an unsupervised approach, a 2-step approach. First, we find major axes of variation in the data, the leaf economic spectrum, hopefully. And then second, we use those axes as new predictor variables in some statistical test. So for our analysis, we used a really common unsupervised approach, PCA, principal component analysis, and then took those axes that were produced and used them as predictors in a linear regression with the precipitation niche as the response. So here's our results. This is a principal component analysis biplot. The observations are points, the variables are arrows on here, and I've plotted the response variable precipitation in color. That first axis, principal component axis, was indeed the leaf economic spectrum. We have variation from resource acquisitive to more resource conservative. So variables like thickness, leaf mass per area, and leaf dry matter content indicate more robust leaves on the right and more delicate leaves on the left. And variables like A and VC max indicate more photosynthetically efficient plants on the left and less so on the right. So this axis captures about 1/3 of the variation in traits. That's the r squared x is about 0.32. And if we go back to our question, is the position along the LES related to precipitation niche? The answer is no. And you can see that just that there's no pattern in the color here on this plot. But also our regression has a very low r squared y, low proportion of variation in the precipitation explained, and it has a high non-significant p-value. But we could take that exact same data set and instead ask, do plant traits vary with precipitation niche? And this is a fundamentally different question. It may sound similar, but it's a different question and it requires a different approach, a supervised approach. And in this case, it's just one step. We find axes that best explain the variation in the response. So for our analysis, we used partial least squares regression with all the plant traits as predictors and the precipitation niche as the response. And this generates p-values through permutation testing, and I'll explain the reason why permutation testing is important a little bit later. So here are the results from the supervised analysis, and in this case, our first axis is not the leaf economic spectrum. We still have this relationship with thicker leaves on the right and less robust leaves on the left, but this first axis is no longer related to photosynthesis at all. Therefore, it captures less variation in the data, so the r squared x is about 0.2. But if we go back to our question, do plant traits vary with precipitation niche? Now the answer is definitely yes. R squared y, the more traditional r squared, is about 0.5, so about 50% of the variation in precipitation is explained by plant traits. And our p-value is low and highly significant. So you might be thinking, did Muir et al. Make a mistake? Did they make the right choice in their analyses? Well, yes, they did. Their original analysis used an unsupervised approach, and so that was correct because it matched their question. If they had used a supervised approach, it would have missed the leaf economic spectrum entirely, and so they made the right choice. But it's also important to realize that their analysis does not answer the question, do plant traits vary with precipitation? That would require a supervised approach. So to further illustrate the consequences of this mismatch between a question and an approach, we did a simulation study. Here we generated data sets with a categorical response, either group A or group B, and 25 predictor variables that were randomly generated under 3 different scenarios. And for each of these data sets, we applied either an unsupervised analysis with PCA followed by logistic regression on those axes, or a supervised analysis using PLS discriminant analysis. In the first scenario, we called it a parent signal because the variables that discriminated between the two groups were also part of the main axis of variation in the data set. And here, as we expected, PCA and PLS performed very similarly. They both found separation between the two groups. They both had a similar r squared x and a very high r squared y. And in nearly 100% of the data sets, found a significant effect of group. In the second scenario, the discriminating variables were not part of the main axis of variation. We called this a hidden signal. And this is more similar to the Muir et al. Data set where there is a relationship with the response variable, but it's not the same as the axis of variation, main axis of variation. So in this case, PCA often failed to find the separation between the two groups, whereas PLS performed essentially the same as it did in the apparent signal scenario. R squared x was similar and a bit lower. R squared y here was very different. So on average, the supervised approach still had an R squared y of about 0.9, which was much higher than the average for the unsupervised approach. And in only about 60% of the data sets did PCA, followed by logistic regression, find a significant difference between those two groups. Whereas with PLS, it found it 100% of the time nearly. So that supervised approach is not really sensitive to the main axis of variation. It doesn't really care about that. And our final scenario is called no signal. There were no discriminating variables and all the variables were uncorrelated. And here, as we would expect, PCA did not find any separation between the two groups because there isn't a real separation between those two groups. But PLS often did find in this score plot visual separation between those two groups. R squared x was low. R squared y, again, was very different. So for the supervised approach, it was often quite high, on average 0.8. And this is why that permutation testing is important. You cannot just take this predictive component and put it into a t-test or a regression. It will always find separation between those two groups. That's what it's trying to do. And the more variables that you have, the more likely that's going to happen just by chance. So it's important to use an appropriate null model for hypothesis testing, which is what permutation testing does. And it finds a significant separation between those two groups in only 5% of the data sets compared to 8% of the data sets for PCA. So very similar. So in fact, it's probably not a good idea to show this plot. It's kind of misleading because it's not significant. So you may be wondering, how big of a problem is this? How often does this mismatch actually happen? Well, we looked at 1 year of ecological literature, and we found that 40% of papers using PCA, in our opinion, actually had a supervised question. And of those 40%, about half of those papers we think could have used a supervised approach out of the box without any trouble. So why is this mismatch so common? One possible reason is that there's this, that ecologists are getting hung up on choosing statistical tests based on their data. And so, choosing tests where their data match the assumptions and so on, over this more fundamental and more biological decision of choosing an approach that matches your question. Another possible reason is simply a lack of awareness. And so for that, I'd like to provide this, I think, really cool teaching example, a data set of cupcake and muffin recipes. This was actually the inspiration for this whole talk and a manuscript that we've submitted to Ecological, looking at whether cupcakes and muffins are in fact different and exploring that through supervised and unsupervised approaches. And I think even if you know just a teeny bit about baking, you'll find these examples really compelling and will really help drive home the difference between these two approaches. So I have a blog post on my website, ericarscott.com, and this entire data set is available on my GitHub if you'd like to use it in the classroom or in a lab meeting or something like that. So thank you all for watching, and I look forward to answering your questions. And I'm happy to answer questions outside of ESA on Twitter at LeafyEricScott. And thanks again.